So my name is David Bonsall. I've been working with Christoph and Tanya and mostly developing sequencing methods for HIV and thinking about some of the upstream components of the pipeline that that Tanya has just presented on. I will, if I remember, spend a bit of time giving some thoughts towards the end on that specific issue around batching and how you use this sort of research tool in a clinical setting. So we're in the business, aren't we, of developing better epidemic interventions that break the chain for endemic and epidemic infections and outbreaks. I tend to think as a clinician that putting aside treatments and vaccines, that much of that comes down to human behavior. And I tend to think of that as ways of influencing human behavior that can maybe population-wide. So in the context of this pandemic, masks, social distancing and lockdown, population targeted, which may be your outbreak responses, and those individual measures where you need that individual-based information to make a change in human behavior, and the classic example of that is contact tracing. So all of this, is orchestrated by the right policy and communication, and that has in turn got to be informed by the right data, which may be diagnostics, testing, epidemiological surveys that are done routinely and in a structured way to account for biases, local knowledge, the existing knowledge of the social network and how diseases are spread, individual diseases may spread around that social network, and we know that sequencing can help with that, and sequencing, especially combined with modeling, as we've seen in PANGEA and the trials that we've been involved with, is a powerful tool. But all our analysis thus far has been retrospective. We've developed important data to inform policy. But when it comes to an epidemic such as coronavirus, I take this quote from Tony Fauci, who shouted this over the airwaves early on in the epidemic: "The virus sets the timeline. You don't, the virus sets the timeline." And this came to the fore with the work that Christoph and I led initially. Now Christoph's leading with the Department of Health and Social Care in the UK around the dynamics and the of transmission for coronavirus, identifying that so many infections were coming from the pre-symptomatic population, so individuals that didn't know they were infected. And that told us two things. First of all, the policies that were implemented internationally were really not going to work without lockdown to stop the spread. And the only alternative was to get smarter and faster about finding people quickly with contact tracing or with mass testing. But we spent the last 8 months thinking about digital solutions to improve the efficiency of that system. More recently, I've been thinking about some of the specific challenges in Africa, again in this pandemic, and I think it's fair to say that in Africa, prevention is everything. With a country like Zambia having only 80 ITU beds in the whole of the country, and also with weaker economies than what we're used to in the UK, that limit options even for lockdowns and furlough schemes. So these smart solutions that we can come up with may have relevance for those reasons, but have to account for the fact that social networks are very different, and have to account for capacity for testing, with very limited. Much has been said about the inequity of or the potential for inequity in vaccine delivery, but not a lot has been said about the inequities of supply for basic diagnostics. And there are, of course, special problems with the existing capacities in terms of the infrastructure and the equipment in areas in Africa, the places that we work, especially in those rural communities. And you have to factor in, of course, that this is a disease amongst many others that stretches that resource. We are engaged with Helen and in Zambia and the London School of Hygiene and Tropical Medicine, and this is a project led by Kwame, which is an extension of the TREAT study, where we'll be doing, where we are engaged in epidemiological surveys of SARS-CoV-2 in the Kabwe district of Zambia. And this just more on this point about testing and the lack of available resources, where you compare the capacity for Zambia, which has something like 500 to 1,000 tests per day, compare that to the 200,000 tests a day that we're doing in the UK, more now, I think. And the fact that so many of those tests in Zambia are positive, this is out of date, but as of the 9th of August, 28% of the tests that were done were positive, suggesting huge amounts of undersampling. And you know, there is infrastructure out there for doing more tests. They have these large high throughput platforms, the Hologic Panther, the Kefi, the Gene Expert, Roche, Cobas. They're all potentially capable of doing an awful lot more, but cannot compete with the international competition for reagents to work those kits. Now, we know how sequencing can help. We're all familiar with the arguments for more molecular epidemiology in PANGEA, with our work in HIV, in terms of the ability to map out the epidemic geospatially and understand the history of the epidemic, the phylogenetic analyses. We can think prospectively about the potential to identify new sources of new outbreaks and characterize those transmission flows, and with that, the factors associated with transmission. And you know for coronavirus, there's real potential there to think about faster ways of identifying super spreading events that you could act on, and to make estimates of incidence and perhaps potentially changing epidemic trajectories without necessarily being able to test enough in your population. The sequence data may help you with that and those policies around it. But and of course it's important that we consider doing more of that in Africa for coronavirus, because this is a pandemic, and we won't understand the pandemic without that global without that global perspective. You know the method that we use. Tanya introduced it in the previous talk. We sequence the nucleic acids, total nucleic acids extracted from samples using a method, for a very simple method for converting that RNA into sequenceable DNA material. We're interested in RNA viruses, although we have other methods to look at the DNA viruses as well. And this was work that Maria-Teresa and I did to optimize the method for getting the most out of your sample, that we then go on to enrich with probes that we can use, for example, the HIV probes. And then you generate all of this data, including the information about transmission, drug resistance, but also the clinically useful information like drug resistance and viral load. And as Tanya introduced, the potential to look at multiple pathogens simultaneously, and in other work that's led by Tanya, looking at other metrics that potentially may have some clinical use and public health use in terms of the timing of transmission and whether or not it's a recent infection or not. And George McIntyre Cockett down here is our principal technician in the lab. He's currently using this method for coronavirus with double that throughput at 762 samples a week at the moment. And actually, this is all out of date. The cost per sample, I've got another slide to show you what our sort of current cost per sample is. So we have the potential to use this method. Can be very fast when we're given enough samples, and can generate all this useful clinical information, as well as the the information that's of value to public health and epidemiology. And in other news, we have the methods now published in the Journal of Clinical Microbiology. We have a protocol that's also in the public domain, and more recently with Sue Eshelman's group, and in a study that Jessica Fogel and myself co-authored, we validated the vSeq methods together with our bioinformatic pipeline DRM-seq for getting clinical drug resistance information, validated that against a commercially available assay, vsIllumina. And in PopArt, we've used this method to characterize the drug resistance in PopArt, and looking at and identify that the relatively high rates of drug resistance in the different groups that we looked at. This is in the population cohort, which is quite a random sample. We used the method that, as it was then implemented, was not the most sensitive assay out there, less sensitive than most commercially available assays, and was giving whole genome information for samples with a viral load, 95% of samples with a viral load over 5,000 copies per mil. So that from a technical point of view, that exposed an area for improvement, because it's important that we're at least as good, if not better, than the commercially available assays. We learned a lot about drug resistance, and that is work that we're writing up at the moment. This is a multiple regression of drug resistance, a high-level drug resistance to first-line therapy, and the associated factors with that. We find that there was those individuals that were recruited later in the trial had higher rates of drug resistance, and what also that meant, if you think about it, was that those individuals who were infected more recently and were therefore more likely to be younger also had more likely to have drug-resistant virus. So you know, so you can see this. So we got the suspicion of an increase in drug resistance in the trial, and with the implication that younger people who have been infected more recently are more likely to harbour drug-resistant virus than older people. And in an analysis that Newton Ateko has led on, we've confirmed this time trend for increasing drug resistance in the different communities of PopArt, divided here by the triplets of the communities. These are the Zambian communities, and by the different arms of the trial, and put that in context with existing literature from previous studies conducted in Zambia to show, similar to what Ravi has shown previously, but here we can actually see that this increasing drug resistance within even within a single trial run between 2016 and 2018. So you get the feel for the kind of application and the potential to generate clinical information from the sequence data as a byproduct of a method that was actually intended for public health epidemiological analyses. And there is a question that I will come back to about how you could potentially do that in a way that delivers you the information, the clinical information that you need in clinically actionable timeframes, while still taking advantage of the cost-effective ways of batch processing for large studies like PopArt and whatnot. So just another thing, this feeds on really from what Tanya was talking about before. The methods that we use is vSeq works by generating libraries of your total nucleic acids in your sample. We have different ways of doing that, and then enriching from those libraries to pull out the viruses of interest. It was noted that in those cohorts that we had ethics to look at other pathogens, that we were seeing significant numbers of hepatitis B reads in those cohorts. And this was an analysis where I took those reads and just assembled whole genomes from them, and we identified 273 partial hepatitis B genomes, with 158 whole hepatitis B genomes plotted on this tree on the right, and you can see they, as expected, fall into the clade E hepatitis B and clade A, as you'd expect for the areas of Africa that we were working in. And this we were a little surprised, because hepatitis B is a DNA virus, and we were using an RNA-based library for this HIV sequencing. I guess our theory is that what we're detecting there is the pregenomic RNA in the samples and the plasma samples. We have one data set where we've looked at the same samples using an RNA-only method and an RNA plus DNA method of generating the libraries, which is the method that Tanya that generated the data that Tanya was talking about. And you can see that actually in those samples, you generally get proportionally more material when you combine the DNA, but surprisingly not, only twice as much more. So it may well be that RNA sequencing is one way of getting hepatitis B genomes. It also makes the point that in every single library that we've generated for PANGEA, if there is, and we are doing work in collaboration with Philip Matthews in Oxford at the moment to look at the sensitivity of this method, but potentially in every single sample we've generated for PANGEA, there is the potential to quantify and sequence, if there are ethics, the hepatitis B genomes within those samples, and there's some important questions, of course, that we talked about at the last PANGEA meeting around that, with the changing ART landscape and the overlap in treatment for HIV and hepatitis B. I mean, I just wanted to chip in with my sort of philosophical point about sequencing and sequencing pathogens. I don't think we sequence pathogens at all. I think we sequence nucleic acids. And the analogy that I'm using here, if you are interested as an astrophysicist in planets, constellations, new galaxies, you don't tend to develop tools that look for those specific planets or those specific galaxies. You tend to design tools that look at the sky. And I think that it is reasonable to take a similar approach to sequencing pathogens. The ideal is to get a good quantitative and qualitative characterization, an unbiased characterization of all the nucleic acids that you can extract from a sample, be that RNA or DNA. I agree with the nuances that Tanya has said about diagnostics, and actually that the process of diagnosis is more than just characterizing what's in there. But I do believe that that is a downstream exercise to a sequence method that can unbiasedly can sequence all the nucleic acids in your sample with minimal bias. More recently, we've worked with the national coronavirus sequencing pipeline, and which has so far generated 62,777 genomes in the UK, and there are about I think 14 centres contributing to this, and in Oxford alone we're contributing to these genomes at a rate of 768 samples per week, and only the Sanger Centre, which has many more staff and sequencers operating on this, is doing more than that. And our plans moving forward for doing this in the UK, and if we can find the opportunity in Africa as well, is to think about how we can take the method that we've got and create a more sustainable capacity, both in the short term for this epidemic, but then ultimately convert that into something that that could be more sustainable longer term and broader in scope, in terms of being able to assist with epidemics, outbreaks, and just routine the routine demand for sequencing in the clinical setting and in public health settings. The initial ambition of including more, getting a broader reach of other pathogens in this epidemic may well help, for example, to guide testing resources. The majority of tests that are done in the UK, for example, are not people with coronavirus, but with other endemic viral infections, rhinoviruses, RSV. And as we go into the winter months, it would be quite useful to know what's going on with those other pathogens, and guide the testing resources and public health resources appropriately. But yeah, we are aiming for this integrated approach to diagnostics and public health, and really the thing that the sort of use case for the whole thing is to think about what that tool is, or what it should have been in Wuhan at the end of 2019, that would have spotted the outbreak sooner and prevented the situation that we're now in. And so I really use that as a sort of model for thinking about what we can develop going forward. And we've made progress in the last year, despite all of the restrictions on our lab and our inability to do much else. Much of this has been worked on through in the coronavirus work, but our cost currently with the reagent only cost is around 16 pounds a sample. With optimization that we have planned, I think that we can get that down to between 8 and 12 pounds per sample. Certainly we can get it to 12 pounds per sample. The sensitivity, I will briefly mention, we can improve tenfold. We have data to show that that's possible. And turnaround times are our weak spot at the moment with the type of sequencing that we do, but we have ideas about how you could reduce that to as little as 24 hours for your sequencing. And then we have systems that we're planning on developing that will give you earlier diagnostics within that pipeline, within a single pipeline, that can give you the positive, negative, quantitative result, viral load, for example, within 2 hours to 4 hours of receipt of a sample. Plastic consumption, reagent consumption are actually just as important, if not more important, than cost at the moment, with stockouts of all of these, and with working on ways of reducing that. And it's important that it's integrated, and it's important that it's a platform that scales. And I think that that's very important. Although I would say that one solution to scaling is not just about thinking of getting a method that can work with sort of single samples, but to think about a method that can do more, such that the throughput that you get into a clinical lab, for be it sort of hepatitis B diagnostics, hepatitis C diagnostics, respiratory virus diagnostics, HIV diagnostics, all goes through the same pipeline. And then your issue about throughput is a bit different. So we have recently trialed the PacBio instrument in Oxford using a version of the vSeq platform, and I'm going to show you some exciting data on that now. Where we used the HIV seroconverters and 96 samples from that. And this was an experiment where we compared to existing Illumina sequencing, the sequencing that you're used to seeing in PANGEA, and we essentially recaptured existing libraries that we generated for our Illumina sequencing, and we sequenced the material on the Sequel 2. And this was in collaboration with Liverpool, who have a PacBio instrument. We're about to get one in Oxford, but this was done in Liverpool. And we did some, and PacBio is useful because it can generate longer reads, longer sequences than what we're used to. So we attempted various ways of improving the size distribution of what we sequence. The PacBio instrument works. It's known to generate long reads. It's also known to generate long reads with error, similar to nanopore sequencing. But the way that it deals with error and the way it improves error is by taking your fragments of nucleic acid that you want to have sequenced, and they can be very long fragments, sticking hairpins on the end of these fragments, and then the sequencing method, which is an enzymatic reaction that will pass through the loop. If you imagine this hairpin now as a loop of a single-stranded molecule, it will go round and round that loop and generate multiple copies of the sequence of interest, and importantly in both direction, antisense complementary direction, so that the sequence of interest is represented by both the sense and the antisense codes. So that deals with any systematic errors that you might have in the sequencing. And without any optimization to increase longer inserts, PacBio just sequencing the libraries that we have for all of the samples really in PANGEA, we were able to demonstrate, and this is the pop art samples, you're able to see a much longer subset of the reads than we do with Illumina, which will only give you insight into the shorter fragments within the libraries that you generate. So whereas for Illumina our insert size is around 350 bases, with the PacBio without any optimization at all, the mean insert size is around 600. With plenty of reads that go out to 15 to 1,500 and even 2,000 bases long. So you can start to imagine, we're all familiar with the phylloscanner type analyses that are restricted by the alignment windows that you can create for your reads, your sequences, and you can start to imagine that you'll get better phylogenetic trees with more resolution if you're able to widen those windows and do some haplotype reconstruction. So this analysis was basically looking at what phyloscanner could do with that that sequence, and on the right you see what we get with Illumina, the sort of numbers of reads on the y-axis that you get with windows of 200 and 250. We actually do a lot of our transmission analysis with very little data at all. First of all, you see that that with the same windows, you're sequencing much. Yeah.
Can you wrap up in the next few minutes?
Yeah, I can. Yeah, and we were able to get similar amounts of data with longer reads for PacBio. So we're just very briefly, this is a ongoing work to increase the size distribution of the libraries that we generate with vSeq. And by increasing the PCR extension times, we predict that that's starting to give us peaks now in the sort of 10 kilobase range, which for HIV would be the whole genome range for sequencing. We've done some work on error. We need to do a bit more, but but a lot is already known about error with PacBio and how you can get with Illumina level qualities. What this does is actually increase the sensitivity of the method, and because you're seeing a greater proportion of the total library, including those longer reads. So on the left, you see that with Illumina, whereas we're only able to get whole genomes with a viral load of 5,000 and above for 95% of the samples, we're starting to see whole genomes now with as little as few as 200 bases, 200 copies per mil. And the ability to link the diagnostics with the sequencing is absolutely key. So one of the problems that you have at the moment that we face, that all labs that have been working in the COG UK consortium in the UK, is that we're left with the remnant scraps of the diagnostic reactions to proceed with sequencing. So the better solution is to think about a single way whereby you generate your libraries, and as a byproduct of generating the libraries for sequencing, you're generating the diagnostic information as well. And so we have developed that method, and we're able to get a PCR estimate of. We know that our method is quantitative here on the left, but what we see here is that the quantitative information that you get from doing your qPCR from the early stage of your library, it correlates quite strongly with the qPCR that you generate from the primary sample. And I've told you that actually the cost of this assay is now, as potentially as cheap as £8 a sample, and so the diagnostic component of that may be around £5 per sample, with an option of going on to sequence at a cost of £8 per sample. The neat thing about this is that it decouples that diagnostic component from your sequencing, so you're able to get the early diagnostic information within a couple of hours and process a few samples at a time, and then batch your samples as necessary for sequencing. So I'll leave it there, and I will leave the discussions about how we're going to bring this together in the Amphius grant moving forward for next week. And I'll just acknowledge the people involved at the Big Data Institute and the Welcome Centre. Paolo has recently joined the team. I'd like to thank all our collaborators in Zambia and the communities, and many thanks.